I'm Defne Baskurt, an 11th grade student at Eyüboğlu Science and Technology High School. Today, I would like to talk about our project, Redefining Food Shelf Life through Microbial Resistance Engineering. Normally, I would be presenting this project with my friend Zeynep Selçuk, but unfortunately, she cannot attend the presentation. Firstly, I would like to explain the inspiration behind our project. Zeynep often falls ill during the winter. Whenever she gets sick, her mother makes her cranberry syrup. After Zeynep shared this with me, we became curious about the properties of cranberries and began our research. The main aim of our project is to extend the shelf life of meat in the refrigerator using bioplastics produced from cranberry extracts. The antioxidant properties of cranberries can prevent microbial spoilage of meat, and if these bioplastics are used in food packaging, they can serve as an environmentally friendly alternative to traditional plastics. Plastics are among the most important products used to enhance the quality and comfort of life today. Since plastics can easily degrade into microsizes, they can cause environmental pollution by spreading into the air, water, and soil. Bioplastics, on the other hand, are polymers obtained from natural sources such as cellulose and starch. These are developed as new generation plastics that do not harm the environment and are derived from plant materials. According to a study, microplastics are found in our bodies due to packaged products we consume because they are so small. What is starch? In this project, we use starch as the raw material for bioplastics. However, starch needs plasticizers like water, vinegar, and glycerin to turn into bioplastic. These materials should be mixed homogeneously and then boiled. After boiling, the mixture should be left to evaporate the water, resulting in bioplastic. These bioplastics have the same texture as market plastic bags. Cranberry. Cranberries can be grown in most regions of Turkey and are highly resistant to cold temperatures. Additionally, they have high antioxidant and vitamin properties, which is why we use them in our project. In this project, we used vinegar, starch, water, glycerin, cranberries, and their extract. During the project process, we made 3 different bioplastics. The first one was normal bioplastic. The purpose of making this bioplastic was to observe the effect of cranberries. While making normal bioplastics, we used starch, glycerin, vinegar, and water. Our second bioplastic was cranberry extract bioplastic, where we used cranberry extract instead of water. Since cranberry bioplastic had not been made before, we had to determine the concentration of cranberries ourselves, so we made bioplastics with a few different concentrations. Our last bioplastic was cranberry pulp bioplastic. In this bioplastic, we used cranberry pulp along with cranberry extract. We added the pulp while extracting cranberry extract back into the mixtures to recycle it. To observe the effect of cranberries, we conducted several experiments and observations. The first was to observe the protection of meat in the refrigerator. We wrapped the meat with bioplastics and kept it in the refrigerator at 5 degrees for 10 days. According to our research, meat spoils in the refrigerator within 5 days, but we waited 10 days to get conclusive results. Additionally, we wrapped one piece of meat in the cling film as it is not commonly used for preserving meat in the refrigerator. After 10 days, we observed color and odor changes in the meat wrapped in normal bioplastic and cling film. However, we did not observe any color or odor changes in the meat wrapped in cranberry extract and pulp bioplastic, and we also saw that it dried out the meat. To observe the bacterial growth inhibition, we used the disk diffusion method. We spread Acinus, E. coli bacteria, and Candida fungus on the media and placed the bioplastics on them, then incubated them at 35 degrees for 5 days. After 5 days, we saw that the largest bacterial growth areas was on cling film and the smallest was on cranberry pulp bioplastic. Additionally, we observed that cranberry had no effect on Candida fungus. Our other method was the bacterial inoculation method. Here, we inoculated bacteria on the samples taken from the bioplastics. The results showed that bacteria grew and colonized the most on cling film and the least on cranberry pulp bioplastic. After applying this method, we integrated technology into our project by using a colony counter application. The application confirmed our results, and here you can see the tables we prepared with the data obtained from the application. In conclusion, we observed that the antioxidant properties of cranberries extended the shelf life of meat in the refrigerator. Additionally, if cranberry bioplastics are used in food packaging, it can help reduce the environmental pollution caused by plastics to some extent. We have a few suggestions for further development of this project. One suggestion is to conduct experiments on different foods. We test on meat, but it can also be tested on other perishable foods like dairy products. Another suggestion is to conduct durability tests. In this project, we focused on extending the shelf life of meat and did not perform durability tests on bioplastics. However, for bioplastic to be used in food packaging, some tests should be conducted. Since we are high school students, we had to use non-pathogenical types of bacteria, but tests should also be conducted with pathogenic types of bacteria. Additionally, we observed that cranberries dry out the meat, which can be an economical method for meat drying since meat drying processes are quite expensive. Thank you for listening.